ada buat sikit-sikit perubahan pada lecture note, okay, supaya saya senang nak refer kepada text and also figure, okay. Tapi content all the same. So yesterday we stopped at number 3, which is cooling by evaporative heat loss. Okay. Okay. So basically, just to recap on yesterday's lecture, which is basically there is 6 ways of how animals regulate their body temperature. So we have looked at insulation. Insulation is the role, for example, a layer of fat in keeping the heat, the body heat within the body, and also you have fur and also feathers, that keep the insulating layer of heat above the skin, okay. And then kalau for example for whale and walruses that lives in really cold area, so they have blubber, right? And then for the second adaptation is on circulatory adaptation. This is basically on the arrangement of blood vessels, where for example you have counter current flow of blood in the artery and vein that flows in the opposite direction, and the heat from the blood that flows in the artery will be transferred to the vein. So this prevents the body heat from losing from the body when the blood reaches at the extremities of the bodies. Okay, extremities such as, for example, the leg or the fins or of the animal. Okay, so another one for circulatory adaptation was, what was the other one? Vasodilation and vasoconstriction. Okay, so vasodilation and vasoconstriction. So basically, your superficial blood vessels, superficial blood vessels are blood vessels that are near the skin. Okay, will vasodilate. So vasodilate is so that more blood will be carried at the surface, near the surface of the skin, and the body heat will be released. Okay, through the vasodilation of the blood vessels. That is to release body heat. Okay, for vasoconstriction is to conserve heat within the body, where blood vessels, especially blood vessels, superficial blood vessels, will vasoconstrict. Okay, to limit the flow of blood, and this is to conserve body heat within the core of the body. Okay, so yang ini akan ingatlah ada dua: vasodilation, vasoconstriction, and the arrangement of artery and vein that allows blood to flow in the opposite direction, which is counter current exchange. Right. So now we are going to look at cooling by evaporative heat loss, behavioral responses, adjusting metabolic heat production, and physiological thermostat. Suara saya dengar perlahan ke dengar jelas? Okey je?
Jelas.
Jelas.
Okay. So next is behavioral responses. Cooling by evaporative heat loss. So many types of animals, such as bird and mammals, lose heat through evaporation of water in sweat. Okay, so we sweat so that the sweat can absorb heat from the body, and when the sweat evaporates, it also releases the heat from the body. Okay. So terrestrial terrestrials meaning that animals that lives on land, okay, mammals, okay. Kita ada aquatic, kita ada terrestrial animals. Okay, aquatic dalam air lah, terrestrial on land, alright. Terrestrial animals lose water by evaporation from their skin and respiratory surfaces. Okay, respiratory surfaces, ada belajar respiration kan? Masa sekolah dulu, respiratory surfaces macam for example for mammals we have lungs kan, for grasshopper they have tracheal system, okay, they have the spherical to lose water through evaporation. So the frogs, okay, the punya respiratory surface can be skin and also lungs. Okay, so evaporation from the skin and respiratory surfaces. So many terrestrial mammals have sweat glands controlled by the nervous system. All right. So panting increases the cooling effects in bird and also many mammals. So panting is when the animals breathe heavily. Okay, they open their mouth and breathe heavily. For dogs, for example, they will stick out their tongue. Okay, when they're panting, so that it exposes the tongue, the surface of the tongue, so that this releases the heat through evaporation. Okay, so next is behavioral responses. So most both endotherms and also ectotherms use behavioral responses to control body temperature. So for example, we have frogs. Okay, so frogs, how does frog controls body temperature is when, for example, when the weather is hot. Okay, so they have to find a much more cooler area, okay, shady area, to cool down their body temperature. Okay. And then as for the dragonfly, they have obsolete posture. So obsolete is like this. And so this is to minimize the body exposure to the sun. Okay, so berbanding kalau dia mendatar je macam ni, so this exposes the body of the dragonfly more to the sun. Okay, the radiation of the sun. But if the dragonfly have this posture, obsolete posture, so this will minimize the body of the dragonfly exposed to the radiation of the sun. Okay. And then the next one is honeybee. So honeybee ni dia ada dua cara dia bergantung kepada keadaan lah kan. So the first situation is when the weather is cold. Okay. So in order to increase heat production, so they will huddle together, thereby retaining the heat. So huddle together tu maksudnya dia akan a cluster. Okay, they will cluster around each other. For example, like this one. Okay, dekat antara satu sama lain. So this is to retain body heat. Macam kita lah kalau sejuk, kalau nampak kucing sejuk ke apa, they will cuddle together so that this will maintain the body heat from losing to the surrounding. So when the weather is hot, okay, so the bee will transport water, okay, to their hives, and then they will fan their wings, kan, dia akan kepak-kepakkan sayap dia, so that dia akan macam buat macam mata kipas kat situ kan. Okay, so itu dalam keadaan hot temperature lah, hot weather. So cool the hives by transporting water to the hives, and then they will fan their wings, okay, to promote heat loss, okay, from the hot temperature. Okay, so the next one is adjusting metabolic heat production. So endotherms control heat production through thermogenesis to match changing rates of heat lost. Okay, so this one is for endotherm. Okay, control heat production through thermogenesis by changing rate of heat loss. So thermogenesis is increased by muscle activity. Okay, so muscle activity such as moving or shivering. Right. So when the muscle contract, for example, this will generate heat. Okay, because contraction of muscle requires metabolic activity within the cell to occur. Okay, through, for example, cellular respiration that produces energy for the muscle to contract, and also heat. Okay, so heat is released. So this will increase the body temperature, will maintain the body temperature. Okay. So, for example, here you have a few large reptiles. Okay, a few large reptiles. Selalunya kalau ular, we know that snake, okay, they are reptiles and they are ectotherms. Okay, but at certain situation, they can be endotherm. Okay, they can be endothermic in certain situation. So, for example, female Burmese python maintain a body temperature of roughly 6 degrees above of that surrounding air during the egg incubation. Okay, so ini kalau untuk phyton ni, in order for the phyton to maintain a warm body temperature to incubate the eggs, dia akan macam, dia tak explain sangat lah Zat. Dia akan macam contractkan muscle dia. Okay, dalam nota ni dia tak detail sikit. Sekejap saya cari nota saya yang lama. Sekejap. Ini. Okay, this one. So this is my old notes, if you could see. Okay, so how does the Burmese phyton maintain a body temperature 6 degrees above the surrounding air? Is that the phyton generate heat through spasmodic muscle contraction. Okay, so by shivering. So the spasmodic muscle contraction of the phyton will increase the body temperature 6 degrees above the surrounding temperature. Okay. So next one is shivering and non-shivering thermogenesis. So for this one, okay, sekejap saya explain yang untuk phyton ni dulu. So phyton, as you can see, during egg incubation, they will increase oxygen consumption. Okay, oxygen consumption increase sebab kalau tahu untuk cellular respiration berlaku, kena ada oksigen lah kan. So oksigen diperlukan untuk cellular respiration, so that during cellular respiration energy is produced, ATP, and also heat is released. So this heat will maintain the body temperature of the snake higher than the surrounding. Okay. So this muscle contraction is a form of shivering and the source of the phyton's elevated body temperature. Okay, so this is how the snake maintains a warmer body temperature compared to the surrounding environment. Okay. So for the next one is shivering and non-shivering thermogenesis. So it says here, mammals and birds in cold environment can increase their metabolic heat production as long as they have adequate food. Okay. So mammals and birds increase their metabolic heat production by as much as 5 to 10 times. Okay, so they have to take more food. Okay, adequate amount of food so that they can make sure that the food can supply enough fuel for cellular respiration to occur to generate heat. Okay. And then next one is flying endothermic insects such as bees and moths have capacity to elevate body temperature. So these insects warm up by shivering before flight. So kalau insects ni they are ectothermic usually kan. Tapi there are endothermic insects when they are about to fly. Okay, flying kan akan menyebabkan badan tu exposed to cold temperature, right? Okay. So to warm up the body, so the insect will increase muscle contraction. Okay, shivering. So shivering before flight. So this is to increase the insect's body temperature. Alright, so back to the old notes. So this is the old notes. Okay, so the next one is the last one is physiological thermostat. Okay. Tak salah. Okay. Okay, so yang tadilah this is on your old notes. Okay, on kita ada thermogenesis kan? Kita ada the next one is non-shivering thermogenesis. So non-shivering thermogenesis, it does not involve physical action. Macam tadi kalau thermogenesis, it involve muscle contraction. Okay, muscle to contract to increase heat production. But the other one is non-shivering thermogenesis. So for non-shivering thermogenesis, this depends on the function of mitochondria. Okay, so it does not involve physical action. So in some mammals, certain hormones can cause mitochondria to increase their metabolic activity and produce heat. Okay, so some mammals have a tissue called brown fat specialized for rapid heat production. So this is non-shivering thermogenesis that does not involve contraction of muscle, but it involves the function of the mitochondria, which is controlled by hormones, to increase metabolic activity to produce heat, more heat. Okay. So together, non-shivering and shivering thermogenesis enable mammals and birds to increase their metabolic heat production by as much as 5 times to 10 times. Okay, so ingat ya, thermogenesis, muscle contraction. Okay, increase muscle contraction. Shivering, for example, non-shivering involve the function of mitochondria, okay, to produce heat through metabolic activity. So the next one is physiological thermostats. So regulating body temperature in humans and mammals is brought by a complex system based on feedback mechanism. So sensors for thermoregulation is concentrated at the hypothalamus, where there is at the hypothalamus a group of nerve cells that function as thermostat. Okay, a group of nerve cells that is located at the hypothalamus. Okay, so we call it as thermostat. So this group of nerve cells will detect the change in body temperature. So thermostat respond to body temperature outside the normal range by activating mechanism that promotes heat loss or heat gain. So kalau ada berlaku perubahan pada suhu badan, terutamanya bila ada perubahan pada surrounding temperature, okay, that may influence the body temperature. So in order to prevent the body temperature from changing too drastically and to maintain the body temperature at the right temperature. So that is the function of the thermostat, to detect any changes, okay, that occurs to the body temperature. So the normal body temperature is around 36 to 38 degrees Celsius. Selalunya kalau sekarang ni kan banyak, pergi mana pun ada thermometer, right, to check your body temperature. So for the normal thermometer tu, selalunya suhu badan yang kurang daripada 37.5, okay, adalah normal. If go beyond 37.5, itu dah considered as demam lah. Okay, so tapi kalau normal body temperature, kalau sihat macam ni, selalunya 36 point lebih-lebih sikit. 36 point lebih-lebih sikit, tak sampai 37 pun. Alright. Okay. So the normal body temperature is this one. So maksudnya kalau macam demam 38 tu okay lagi. Tapi kalau dah 39 ke 40 tu dah beyond 40 cannot. So itu dah macam dah boleh dah sangat critical lah. Maksudnya you have to prevent your body temperature from reaching 40 degrees when you have fever. Okay, kalau tak boleh sawan kat situ. Okay. So normal body temperature increase such as when exercising or in hot surrounding. So this is detected by the hypothalamus. thermostat in the hypothalamus. So the brain will send corrective mechanism, okay, corrective or cooling mechanism to the sweat gland. Okay, so this will cause the sweat gland to secrete sweat, and through the evaporation of sweat, it removes body heat. Okay, and also another instruction that comes from the brain, from the hypothalamus. Okay, so it causes the blood vessels to vasodilate. Okay, blood vessels in the skin to vasodilate. So when the blood vessels dilate, so it brings more blood, okay, to the skin, and releases the excess heat from the body through radiation. Okay, heat radiates from the skin surface. So this will bring back the body temperature, decrease back the body temperature back to normal. Okay. So ingat ini adalah dua cara macam mana badan awak release heat, okay, from the body, right? So when the body temperature, for example, decreases below the normal range, such as in cold surroundings, so it is again detected by the thermostat in the hypothalamus that activates warming mechanism. So it sends the corrective mechanism to these two structures, which is the skeletal muscle. So this causes the skeletal muscle to contract rapidly, okay, causing shivering. So rapid contraction of the muscle cell generate heat. Okay, so it increases cellular respiration. Cellular respiration increases the production of ATP so that the muscle can contract rapidly, and also that process also releases heat, okay, to warm the body. And the effects to the blood vessels is that it causes the blood vessels in the skin, okay, near the skin, to constrict. So vasoconstriction. So this will divert the flow of blood from the to the skin to the deeper tissue. Okay, so this reduce heat loss from the skin surface. So thus, body temperature will increase to the normal range. Okay. So that's all the 6 adaptation of how animal maintains their body temperature. Okay, thermoregulation. So now we are going to look at the next negative feedback by the body, which is osmoregulation. Okay, osmoregulation of in the human body. So for this part, we are going to look at the homeostatic organ, which is for this one in order to control a stable fluid concentration within the body. Okay, pH concentration within the body fluid. So this is done by homeostatic organ, which is the main organ is the kidney lah, right? And then we are going to look at the formation of urine and the role of these two hormones, which is ADH, antidiuretic hormone, and also aldosterone. Okay, so kidney. Kidney, as you know, it is the homeostatic organ. Okay, homeostatic organ. So the excretory organs, it is the excretory organs of vertebrates function in both excretion and osmoregulation. So kidney has these two main function: excretion, to excrete nitrogenous waste in the form of urea. Okay, so this is involve the formation of urine later. And then osmoregulation, to control solute concentration and also water balance in the body fluid, water gain and loss. Okay. So the mammalian kidney has two distinct regions, which is the outer renal cortex, the outer part of the kidney, and then in the inner renal medulla. So the inner renal medulla, it is divided into two portions. Later we are going to look at the detailed structure of the kidney. So this is the main region, the two main region, cortex luar, medulla dalam. Okay, so waving back and forth across the renal cortex and medulla are the nephrons. So nephrons are the functional units of the vertebrate kidney. So nephrons lah yang akan terlibat dalam reabsorption of nutrients, okay, back to the blood flow, okay, and also the production urines, okay, through the process of filtration that occurs in the nephrons. So part of the kidney, so here these are the parts of the kidney. So you have the renal cortex, which is the outer part of the kidney. It is a smooth, it has a smooth texture. So it is the location of the Bowman capsule and also the glomerulus, in addition to the proximal and distal tubules and the associated blood supplies. So the region, so this is the region of the cortex. Okay, cortex, bahagian luar. Kat sini lah cortex, renal cortex. So this is where glomerulus, kan, glomerulus, proximal tubule, distal tubule are located. Okay, and also blood vessels that surrounds the distal and also the proximal tubule. So nephron ni sebenarnya dia terdiri daripada two main compartment sebenarnya. Satu adalah tubules and second adalah blood vessels. Okay, nephron. Ingat nephron, it is consists of two major structures. One is the tubule, the other one is the blood vessels that surrounds the tubule. So the tubule will start with the Bowman capsule. Okay, so tubule tu adalah lumen yang akan menghasilkan filtrate yang akan menjadi urine later. Glomerulus adalah sebahagian daripada blood vessels that carries blood lah kan. So glomerulus and then kita ada dia akan bersama dengan blood vessels that surrounds the distal and also proximal tubule in the cortex area. Okay, so kalau tengok structure nephron ni, bahagian tubule dia start dengan Bowman capsule, proximal tubule, loop of Henle, okay, descending and also ascending. We have the distal convoluted tubule and then it enters into the collecting ducts. Sebenarnya collecting ducts is not part of a nephron. Okay, collecting ducts, it collects filtrate that is produced, okay, from all the processes that occurs in the nephrons. Okay, so the filtrate from several nephrons will enter into the collecting ducts before it enters into the next region, which is the renal pelvis. Okay, itu structure nephron sebenarnya, dia tak termasuk collecting ducts. Collecting ducts is where it collects the filtrate from several nephrons. Okay. So that is okay. That the first region adalah renal cortex. Okay, bahagian luar. This one. Okay, renal cortex. And the inner part is the renal medulla. So renal medulla ni dia terbahagi kepada dua region. The outer medulla and also the inner medulla. If you look at this diagram here, I hope that you have watched the videos, okay, explaining the process. So medulla ni dia terbahagi pada dua, outer medulla and inner medulla. So atas ni adalah renal cortex, outer medulla, inner medulla. Okay, so these are the region. Kalau tengok gambar ni so this is the cross section of kidney to illustrate a nephrons. Okay, a nephron. So you have the cortex, outer region of the kidney, medulla. So medulla is separated into two regions, outer and inner medulla. Okay, so for the cortex, it is where the glomerulus located, the Bowman capsule, the proximal tubule and also the distal tubule will be located. As for the outer medulla and inner medulla, it is where the loop of Henle is located and also the collecting ducts. So as you can see, collecting duct yang cabang-cabang ni adalah cabang daripada several other nephrons. Okay, so the filtrate from several nephrons will enter into the collecting ducts before it enters into the renal pelvis. Okay. Okay, so renal pelvis funnel, it is a funnel shaped basin that receives urine drained from the kidney nephrons, okay, so from all the nephrons within the kidney via collecting ducts, okay, and then to the larger papillary ducts. Okay, so kalau awak tengok kat sini, so this is the renal pelvis. Okay, so the renal pelvis will collect the urine formed, okay, from all the nephrons here, that the nephrons will convey the urine produced through collecting ducts and then into the renal pelvis and then into the ureter. So the ureter will then lead the urine to the urinary bladder before going out through the urethra. Okay. So kalau so this is the structure of kidney. Okay, so you have to really label all the structure, especially when you are drawing your kidney structure in your lab report later. Okay, so all the structures that you label will contribute to marks. Missing one of the structures will cause you to lose mark. Okay. So here you have the two blood vessels here. So you have the renal artery that brings oxygenated blood, okay, oxygenated blood to the kidney, and also you have the renal vein, okay, that removes the oxygenated blood from the kidney. Okay, so nephrons ni ada satu jenis nephron, ada nephron ni dia ada dua jenis sebenarnya. So the nephrons that has a long loop of Henle. As you can see here, for nephron that has long loop of Henle, we call it as the juxtamedullary nephrons. And another type of nephron is cortical nephrons. Cortical nephron will have shorter loop of Henle. Pernah keluar dalam soalan, saya tak ingat, dia macam lebih kurang macam ni lah. Just what is the significance of juxtamedullary nephrons, okay, to maintain in for animals, okay, for animals that lives in hot area. Apa signifikan juxtamedullary nephron pada haiwan yang duduk tempat yang panas? Obviously, for animals that live in hot area, they have to conserve water in their body. So in order to conserve water in their body, by having long loop of Henle, okay, so by having long loop of Henle, as you know, at the descending limb of loop of Henle, it is where reabsorption of water occurs. Okay, so by having long loop of Henle, more water will be reabsorbed. Okay, more water will be reabsorbed, and for that animal, because more water will be reabsorbed into the body to conserve water in the body, so for that animal, they will produce very concentrated hyperosmotic urine. Okay, itu antara soalan applied yang dia boleh keluar lah. So you have to really understand the process. Sebab bila soalan keluar, apa-apa soalan pun yang keluar dalam exam nanti, you can apply your knowledge to that question. Okay. So berbanding dengan cortical nephrons. So cortical nephrons will have shorter loop of Henle. So when you have shorter loop of Henle, less water will be reabsorbed. Okay, so this will produce more diluted or hypoosmotic urine. Okay. So right. So it says here, the mammalian excretory system enters a paired kidneys, okay, enters on paired kidney. So we have two kidneys, a pair of kidney, so which are also the principal site of water balance and salt regulation. So each kidney is supplied with blood by a renal artery and drained by renal vein. So renal artery will bring oxygenated blood to the kidney, and through the renal vein it removes deoxygenated blood. So urine exit each kidney through a duct called the ureter. Okay, so from the kidney to the urinary bladder is through the ureter. So from the urinary bladder to the outside of the body, so it's through the urethra. So don't confuse between these two terms. Ureter dulu baru urethra. Okay. So as you can see, ureter first. Okay, ureter is between the kidney and also the urinary bladder. Okay, and then out from the body through the opening of the urethra is the urethra lah, kan? So it conveys the urine out of your body. So as you can see, so these are the two kidneys. Okay, so two kidneys, the right kidney and also the left kidney. Terbalik the right kidney and also the left kidneys. So above the kidney, so this is you have the adrenal gland. So later in the subsequent chapter, so we are going to look at this chapter of hormones. So you are going to learn that adrenal gland is the gland, okay, that produces hormones that regulate certain metabolic activities in your body. Okay, kita ada two different regions within the adrenal gland. Okay. So here you have the aorta, okay, aorta, the large blood vessels here. Mana dia punya labeling dia? Okay. The abdominal aorta, okay, abdominal aorta will bring oxygenated blood. So here it enters into the artery, renal artery, and then enters into the kidney, okay. So ini dia akan branch lah. Left renal artery, right renal artery, okay, to supply blood to the kidneys. And then you have the inferior vena cava. Okay, inferior vena cava is the blood vessels that bring deoxygenated blood from your body to the heart. Okay, to the heart, back to the heart, back to the right atrium. Okay. So here from the vena cava, so they can branch to these blood vessels, which is the renal vein. Okay, the renal vein, the right renal vein, the left renal vein that removes poor deoxygenated blood back to the heart. Okay, so the nephrons. So the nephron consists of a single long tubule and a ball of capillaries called the glomerulus that filter blood. Okay, that it filters blood. So basically, as I've mentioned, nephron, dia ada dua region. Okay, the tubule and also the blood vessels. Okay, blood vessels start dengan glomerulus. Alright, so actually glomerulus is a ball of blood capillaries. Okay, so start of the tubule adalah Bowman capsule. So Bowman capsule, it surrounds the glomerulus. Okay. So Bowman capsule surrounds and receives filtrate from the glomerulus. Filtrate ni adalah fluid that enters all the way along the tubule. Daripada Bowman capsule ke proximal tubule, ke loop of Henle, ke distal tubule, ke dalam collecting ducts, all that fluid we call it as filtrate. That will become the urine later. Okay, filtrate. Ingat istilah tu, okay? So filtrate is the fluid in the tubules, starting from the Bowman capsule, proximal tubule, loop of Henle, distal tubule, collecting ducts before it comes out as urine. Okay. And then starting of the blood capillaries is the glomerulus. Okay, so glomerulus and then out into the into the blood vessels that surrounds the proximal and also distal tubule. Okay, so this is the glomerulus. So from the glomerulus, as you can see from this diagram here, so this is the renal artery. Okay, renal artery. Okay, and then it enters into the glomerulus. So the blood vessels that enters into the glomerulus here, we call it as the afferent arteriole. Okay, afferent arteriole enters into the blood vessels in the glomerulus and then out to the efferent arterioles. Okay, so you have to take note. You have to really take note here. So this is really significant. It is really for you important for you to understand. Afferent arteriole, it has diameter bigger than the efferent arteriole. So what is the significance of having bigger diameter for afferent arteriole compared to efferent? So when blood, as you know, blood that flows in the artery has higher blood pressure. Okay, so when blood flows at high pressure at the afferent arterioles, and then dia kena keluar pula dekat blood vessels that has that has a smaller diameter, which is the efferent. So this will create a high blood pressure or a high hydrostatic pressure in the glomerulus. So this pressure, okay, the high hydrostatic pressure or the high blood pressure in the glomerulus will cause the fluid in the glomerulus, the excess fluid to go out, okay, to diffuse out of the glomerulus and enters into the Bowman capsule and becomes the initial filtrate. Okay, it becomes the initial filtrate. So it says here, the Bowman capsule surround and receive filtrate from the glomerulus, and then after that you have the proximal tubule. Okay, proximal tubule. So this is the proximal tubule. Ingat dia punya sequence dia. Okay, it is really important to memorize, to know the sequence of the nephrons, the structure of the nephrons. So after the Bowman capsule is the proximal tubule. Okay, after the proximal tubule is the loop of Henle. So for the loop of Henle, maintains concentration gradient. Tadi saya lupa, proximal tubule is where reabsorption of water occurs, salt, glucose and amino acid occurs. Okay, reabsorption maksudnya daripada filtrate masuk balik ke dalam blood vessels. Okay, reabsorb. Your body reabsorb these valuable substance, water, salt, glucose and amino acid. You do not want glucose and also especially amino acid to be in your urine. So this substance has to be reabsorbed back into your blood circulation. Okay, so after proximal tubule is the loop of Henle that maintains concentration gradient, and then after that it enters into the distal tubule. It is the tubular secretion of hydrogen ion, potassium and certain drugs. These are the substance that needs to be removed from your body. So they has to be in the filtrate. So whatever substance, kan, hydrogen ion ke, potassium or drugs or any other substance that need to be removed from your body will end up in the filtrate through the process of secretion. Ingat reabsorption daripada filtrate pergi masuk balik dalam darah, secretion daripada darah masuk ke dalam filtrate untuk jadi urine later. Okay, so these are the two terms that you have to really know where to use. Okay, secretion and reabsorption. Okay, so this is the structure of nephron lah, as like I've explained. Okay, so this is nephron. It has two major region, the tubule yang warna kuning, okay, and the blood vessels. Tubule start from the Bowman capsule, proximal tubule, loop of Henle, distal tubule, and then it enters into the collecting ducts that collects filtrate from several other nephrons. And as for the blood vessels, it starts with the glomerulus, and then you have the blood vessels that surrounds the proximal and the distal tubule. We call it as the peritubular capillaries. Tengok ni, tengok dia punya diagram. Peritubular capillaries are the blood vessels that surrounds proximal tubule and also distal tubule. Okay, so you have another type of blood vessels that surrounds the loop of Henle. We call it as the vasa recta. Okay, so those are the component of nephrons. It consists of blood vessels and tubules. Okay, so formation of urine. So nephron is organized for stepwise processing of blood filtrate. So the process of urine formation will undergo these three major processes, which is filtration or ultrafiltration, where high blood pressure, okay, will cause formation of filtrates to produce, okay, pressure filtering body fluids. Body fluid is basically from the blood flow, okay, from the blood flows that enters into the glomerulus, okay. So the high blood pressure in the glomerulus due to the different diameter of the two afferent and efferent arteriole will cause a high blood pressure in the glomerulus that causes excess fluid, okay, excess fluid to enter into the Bowman capsule along with other nutrients. Okay, other nutrients such as glucose and also vitamins and also amino acid. So those valuable substances, okay, and also water has to be reabsorption. So reclaim valuable solutes, okay, water, amino acids, vitamins, and glucose. Okay, has to be reabsorbed, and also sodium ions. So this is reabsorption. Reabsorption daripada filtrate masuk ke interstitial fluid, masuk ke aliran darah balik semula. For secretion is involved in the formation of the filtrate. Okay, so secretion add toxin and other solute from the fluid, from the blood flow, okay, into the filtrate in the tubules. Okay, so kidney will remove metabolic waste products such as urea, creatinine, and also uric acids, and secrete them in the form of urine. Okay, so these are the three processes, major processes. Filtration, it occurs here. Okay, glomerular filtration. It involves the glomerulus and also the Bowman capsule. And then second is the tubular reabsorption and tubular secretion. Tubular reabsorption is where valuable substances will be reabsorbed. Okay, and then secretion, any substance from the body fluid that is not needed, okay, will enter into the filtrate, okay, from the blood vessels. Okay, so next is the tubular reabsorption and also tubular secretion at the distal tubule. Mula dekat proximal tubule, proximal tubule after the Bowman capsule. Okay, distal tubule before enters into the collecting ducts. So that will occur also reabsorption and also secretion. Reabsorption of valuable solutes and secretion of waste. Okay, that will end up into the urine. So and then masuk ke dalam collecting ducts lah. So in the collecting ducts region, so akan berlaku reabsorption of water. So when water is reabsorbed, okay, so here you have the collecting ducts. In order to increase reabsorption of water along the wall of the collecting ducts, okay, along the cells of the collecting ducts here, we call it as the transport epithelium. There will be many aquaporins. Okay, so the aquaporins, kita dah belajar dulu masa chapter 3 sem lepas. You have aquaporins, right? So aquaporins are protein channels that increases the transportation of water through osmosis. Okay, through osmosis. So aquaporins allows water to be reabsorbed. Okay, daripada filtrate masuk balik ke interstitial fluid and enters into the blood vessels. Okay, back into your blood circulation. And then you will form concentrated urine here. Okay, depends on your body. Your body depends on the situation. You can produce hyperosmotic urine or hypoosmotic urine. Hyper concentrated urine, hypo is diluted urine. Okay, and then to the renal pelvis, and then enters into the ureter, urinary bladder, and then out through the urethra. Okay. So for the first stage is the filtration. Okay, filtration of blood. Or the glomerular ultrafiltration. You can write either terms. Ultrafiltration boleh, filtration pun boleh. Okay, so at the Bowman's capsule, so the Bowman capsule, it envelopes the glomerulus. Okay, it surrounds. Boleh guna dua istilah, envelopes or surrounds. Okay, the glomerulus. So urine formation occurs here. Okay, it begins here. So blood flows through the glomerular capillary under high pressure, forcing more than 10% of the plasma out of the capillaries into the Bowman capsule. Capillaries is the glomerulus, okay? So the high blood pressure in the glomerulus will cause excess fluid, excess blood plasma, that becomes the interstitial fluid. Sorry, that becomes the filtrate, okay, and enters into the Bowman capsule. So filtration occurs as high blood pressure or hydrostatic pressure forces fluid from the blood in the glomerulus into the lumen of the Bowman capsule. Okay. So ingat kenapa boleh berlakunya high blood pressure disebabkan oleh the diameter of the afferent arteriole is bigger compared to the diameter of the efferent. So as you know, afferent arteriole will carry high blood pressure, okay, at a high velocity and enters into the into the glomerulus and out through the efferent. So this creates a high blood pressure that forces the 10% fluid in the plasma to enter into the Bowman capsule and becomes the filtrate. Okay. Okay, so filtration sini dah. Okay, so factors contribute to the production of glomerular filtrate. Okay, glomerular filtrate of fluid from the glomerulus that enters into the Bowman capsule. That is glomerular filtrate. So hydrostatic blood pressure in the glomerular capillary is high than the other capillaries. Okay, which is the surrounding blood capillaries, the nephron. So high pressure due to high resistance to outflow presented by the efferent arteriole, which has a smaller diameter than the afferent, the afferent arteriole. Okay, which has you better change, which has a smaller diameter than the afferent arteriole. Okay. So a large surface area for filtration provided by the highly coiled glomerular capillaries. So that is the purpose of having a highly coiled glomerular capillary. So it presents a high surface area so that more filtrate can be formed in the Bowman capsule. Okay. So number 3, great permeability of glomerular capillary due to numerous small pores between the endothelial cells, making the glomerular capillary more porous than typical capillaries. Please take note that this is one of the favorite questions that will come out. Okay, that will usually come out during the exam. Okay. So selalunya soalan exam akan tanya, okay, what is the characteristics of the glomerulus? Okay, satu adalah it has many pores. It has numerous small pores. Okay, to increase its permeability, it has large surface area. Okay, because it is highly coiled, and then presence of high hydrostatic pressure. Okay. So ini adalah antara soalan yang suka keluar lah. So when the glomerular capillary is porous, so it allows more fluid from the blood capillaries from the glomerulus to enter into the Bowman capsule. Tapi sebabkan pore yang present dekat glomerulus ni boleh menyebabkan other substance to enter also into the filtrate, which is glucose, amino acids and vitamins also to enter into the filtrate that enters into the Bowman capsule. Okay. So the blood is carried by the afferent arteriole to the glomerulus. So the blood is filtered as the blood pressure forces fluid through the porous capillaries, through the porous glomerular capillaries, walls and also the filtration slit. Presence of this structure, filtration slits. Okay, so this allows fluid from the glomerulus to enter into the Bowman capsule to become the fluid, to become the filtrate. Okay, so the glomerular capillaries is permeable to water obviously, and other small dissolved molecules such as salt, glucose, amino acids, vitamin, urea, and also nitrogenous wastes. Okay. This is another favorite question. Okay, so you have to know lah what are the substance that enters into the Bowman capsule. Okay, from the glomerular capillaries. So glomerular capillaries not permeable to blood cells. Okay, it is not permeable to blood cells, and also large molecules such as plasma proteins. Dia pores yang ada pada glomerular capillaries ni hanya akan bagi small dissolved molecules saja yang masuk dalam Bowman capsule as filtrate. Tapi kalau blood cells, okay, especially red blood cells and also blood plasma protein, dalam blood plasma ada protein kan, macam all other proteins that involve that maintains a stable fluid concentration in the blood. Okay, so those substances will not enter into the Bowman capsule. Okay, so blood move out into the efferent arteriole. So remember, afferent has a bigger diameter compared to efferent, that creates a high blood pressure in the glomerulus. Okay, that forces fluid to enter into the Bowman capsule as filtrate. Okay, so these are already mentioned. So you have a special cell here which is we call it as the podocyte. Okay, so podocyte ni are cells in the wall of the Bowman capsule contact that is in contact with the capillaries of the glomerulus. So this is the podocyte. Okay, cells in the wall of Bowman capsule that is in contact with the glomerulus. And then this is the filtration slit, and then this is the porous glomerulus. Okay, porous glomerulus. So due to the high pressure, small solutes will exit the glomerulus and enters into the Bowman capsule through this filtration slits. Okay, which is a narrow gap here. Okay, narrow gaps. So itulah dia. Okay, so this is the endothelial cell of the blood of the capillaries. Nanti kalau kita belajar circulatory system, structures of blood vessels. So all the blood vessels, artery ke vein ke blood capillaries, bahagian dalam ni kan, we call it as lapisan dalam blood vessels, so we call it as the endothelial cells of the capillaries. Okay. So filtration membrane of the kidney. So the porous wall of the glomerular capillaries and the filtration slits between the podocytes form a filtration membrane. Okay, so combination of this structure, porous glomerulus and this structure, filtration slits, okay, will form the filtration membrane. Okay, so this allows the formation of filtrate that enters into the Bowman capsule later. Okay, so the membrane is highly permeable to water, small molecules such as amino acids and also glucose and also vitamins and ions, but restrict the passage of blood cells and large molecules such as plasma proteins. So plasma protein red blood cells memang maintain je dalam glomerulus. Okay, sebelum dia keluar ke dalam efferent arteriole. Okay, so pathway of the filtrate. Ingat filtrate is the term that we use for fluid that enters into starting from the Bowman capsule along the tubule, along the proximal tubule, loop of Henle, distal tubule, and also collecting ducts that will become the urine later. Okay. So for the loop of Henle, it has two regions. Descending loop of Henle and then ascending before it enters into the distal tubule. Awak boleh jawab, awak boleh guna mana istilah pun boleh. Proximal tubule ataupun nak rajin proximal convoluted tubule. Okay, because it is highly folded. Okay, convoluted. Like this. So it is folded. So that's why we call it as convoluted. Okay. So this is another diagram that you could refer to your as the structure of the nephrons. Saya lagi suka tengok gambar rajah ni sebab dia lagi clear berbanding dengan ni. Kan, berbanding dengan ni you don't really see the dia punya turutan tubules tu. So dia nampak berselirat kat sini kan. Tapi kalau nak mudah, senang nak tengok dan fahamkan structure nephron is by looking at this diagram. Ini saya lagi prefer lah. So here you have the afferent arteriole that has bigger diameter compared to the efferent here, and then it brings oxygenated blood that enters into the glomerulus. Highly coiled blood capillaries, kan? So the glomerulus is surrounded by the Bowman capsule. So Bowman capsule is the first portion of the tubule for the nephron, and then it enters into the proximal tubule, and then it is highly folded, convoluted, okay, and then it enters into the descending limb of loop of Henle, and then up into the ascending limb loop of Henle, and then into the distal tubule, and then into the collecting ducts. So ini yang nampak ni adalah portion of other nephrons. Okay. So maksudnya collecting ducts ni will collect urine from several other nephrons. So surrounding the tubules you have the blood vessels. So blood vessels that surrounds the proximal tubule and the distal tubule, we call it as the peritubular capillaries. Blood vessels that surround the loop of Henle, we call it as the vasa recta. Okay, vasa recta. So for vasa recta, dia pun ada dua region juga. dia ada descending vasa recta, dia juga ada ascending vasa recta. Kat sini kita akan baca nanti akan melibatkan counter current juga. Okay, the blood flow in this region, okay, the blood flow in the descending vasa recta will flow in the opposite direction as the filtrate in the ascending limb of loop of Henle. The blood flow in the ascending vasa recta will flow in the opposite direction as the filtrate in the descending limb of loop of Henle. So this opposite direction of blood flow and the filtrate, okay, will will give significant effects to the water and also solutes being reabsorbed and secrete, okay, into the filtrate. Okay, so kita tengok after the proximal tuber tadi, after the Bowman capsule, kita dah fahamkan apa yang jadi kat Bowman capsule. So next is the proximal tubule. Okay, so proximal tubule of proximal convoluted tubule is the longest and the widest part of the nephron. So molecules are transported actively and passively from the filtrate into the peritubular capillary. Molecules are transported actively and passively from the filtrate. Filtrate tu daripada mana tadi? Bowman capsule kan. Next it enters into the proximal tubule. So ada substance yang reabsorb, ada substance yang secrete. You tengok kat sini you all tengok arrow yang kat atas ni, arrow yang keluar daripada filtrate, itu adalah proses reabsorption. So it means that all these substance, bicarbonate ion, sodium chloride, water, nutrients such as amino acids and also glucose will be reabsorbed back into your blood, and also potassium ion. The substance that will be secreted into the filtrate will be hydrogen ions and also ammonia. Okay, so hydrogen ion, you don't want excess hydrogen ion in your blood. Why? Because this will cause your blood to be acidic. So the excess hydrogen ion has to be removed from your blood and excreted through the formation of urine. Okay, so secrete masuk ke dalam filtrate jadi urine nanti. Alright, so kalau yang arrow merah ni, so those are the substance that moves out of the filtrate, for example into the blood circulation through active transport. Okay, nutrients, structure nutrient kan berbesar kan, amino acids and also glucose. So those will be reabsorbed back into your blood vessels, enters into your blood circulation through active transport. Okay, it means that it has to have transport protein lah. Kan, in order for those substances to enter back into your blood vessels, it has to go through transport protein, through active transport. But for these molecules such as, for example, water, okay, they can diffuse or through facilitated diffusion. Okay, facilitated diffusion also can. Okay, so those are the way of how different substances are being reabsorbed and also secreted. Okay, from the filtrate. So it says here, sodium chloride in the filtrate, sodium chloride in the filtrate enters the cell of the transport epithelium by facilitated diffusion and also co-transport mechanism. If you look at this term, tolong highlight kan, transport epithelium ni. What is transport epithelium? Transport epithelium dia adalah nama sel, okay, it is the name of the cell that makes up the wall of the tubules. Sepanjang-panjang proximal tubule, loop of Henle, kan, distal tubule and also collecting ducts, sel yang menjadikan tubule ni, dia terdiri daripada sel yang kita namakan dia sebagai transport epithelium. So dekat cell of the transport epithelium ni akan ada contohnya transport protein that helps in the transportation of sodium chloride. Maksudnya kat sini akan ada protein, transport protein untuk terlibat dalam transportation of sodium ion and also chloride ion. Facilitated diffusion, okay? So kalau kat sini awak tengok walaupun kat sini dia tunjuk active transport, sodium chloride ada terdiri pada dua ion, sodium ion and also chloride ion. Sodium ion, okay, kalau dicakap kat sini, you look at dia. So the epithelial cells actively transport sodium ion. Sodium ion akan keluar melalui active transport, tapi kalau chloride ion adalah melalui passive transport. Okay, along the wall of the proximal tubule, along the transport epithelium. Okay. So it says here as salt, as salt move from the filtrate into the interstitial fluid. Interstitial fluid is the fluid surrounding the tubule and also the blood capillaries. Okay, so water flows by osmosis and thus reducing filtrate volume. Okay, so lebih banyak the more sodium chloride diffuses out or move out of the filtrate into the interstitial fluid, so the surrounding interstitial fluid will be very concentrated. Will be concentrated. So if the interstitial fluid is concentrated by the sodium chloride, so this will cause water to diffuse out from the filtrate into the interstitial fluid, and causes the filtrate concentration to be reduced before it enters into the loop of Henle. Okay. So glucose, amino acids, potassium and other essential substances are also actively or passively transported from the filtrate to the interstitial fluid and then into the peritubular capillary. So remember, peritubular capillary are the blood vessels, okay, blood capillaries that surrounds proximal tubule and also distal tubule. Okay, so these are the substance that will be reabsorbed. Reabsorb maksudnya masuk balik ke dalam blood flow. Okay, blood flow in the peritubular capillaries. So some toxics such as drugs and toxin processed from the liver are secreted into the filtrate by active transport. Secrete tu maksudnya daripada blood into the filtrate to be removed in the urine later. So these are needs to be removed. Okay, so they are secreted into the filtrate by active transport. Okay, so processing filtrate in this region helps maintain a relatively constant pH. Proses yang berlaku dalam proximal tubule ni juga akan membantu mengawal pH dalam body fluid awak. Dia akan membantu mengawal blood pH. Okay, blood pH. And body fluids lah. Body fluids can be your blood, your lymph and also your interstitial fluid. So those are your body fluid, blood, interstitial fluid and lymph. Okay, body fluids. So so you know that the normal pH for your body is 7.4. Okay, so to prevent your blood from becoming acidic, okay, due to the presence of excess hydrogen ion in the blood, so the hydrogen ion needs to be secreted. Okay, need to be secreted into the lumen of the tubule to become part of the filtrate. Okay, so this is done through the movement of hydrogen ion through the transport epithelium, cells of the transport epithelium. So these cells juga, cells of the transport epithelium ni, right, is also capable of synthesizing and secreting ammonia. Okay, so maksudnya lagi banyak hydrogen ion secrete into the filtrate, it means that the transport epithelium will produce and secrete more ammonia. Okay, so the function of this ammonia is to act as buffer to trap hydrogen ion in the form of ammonium ion NH4+. Okay, so ammonia NH3 kan, so bila dia attach dengan excess hydrogen ion ni, dia akan jadi ammonium ion. Alright, so ammonia is NH3, bila dia gabung dengan H+, secreted into the filtrate, it becomes ammonium ion NH4+. Okay, so it says here, the more acidic the filtrate is, the more ammonia the cell produces and secrete. The cell is the cell here, cells that makes up the proximal tubule, which we call it as the transport epithelium. Okay, so the tubule also reabsorb about 90% of the buffer bicarbonate ion from the filtrate. So from the filtrate, dia nak absorb this valuable ion which is sodium bicarbonate. So sodium bicarbonate is really important because this prevents your blood from becoming acidic. Sodium bicarbonate dia bertindak sebagai buffer jugalah. So this is to maintain a constant pH within the body, fluid within your blood, for example, from becoming too acidic due to the presence of excess hydrogen ion. So yang ini kita akan tengok masa chapter basically respiratory system lah, the function of bicarbonate ion. Alright. And then the filtrate volume decreases and materials to be secreted become concentrated. So as all these substances being reabsorbed, okay, air especially, bila air lebih banyak keluar dekat region proximal tubule, as you know, proximal tubule is the longest part of the nephron. So this causes the filtrate to be more concentrated. Okay, more concentrated, become concentrated lah due to especially reabsorption of water. So next is what happened in the loop of Henle. Kita take a break 3 minutes sekejap. Boleh?
Boleh.
Okay, 2 minit lah. Okay, so kita sambung semula. Okay, so at the region of loop of Henle, so if you look at this diagram, so saya mergekan teks dengan diagram. So in your lecture note, kan, so you can refer to your diagram also. Okay, so sebab senang, as you have text, you have the diagram. Bila belajar bio, jangan read text je tanpa rujuk diagram sebab nanti awak tak faham. Awak baca menonong macam tu kan. You have to have your text and also the diagram to help you understand better of what you are reading. Okay. So loop of Henle, there are two regions. It has two regions, which is first from the proximal tubule, it will enters into the descending limb of loop of Henle. So this is where reabsorption of water occurs. Okay. Okay. So along the descending limb of loop of Henle, only reabsorption of water occurs. Okay. This is why, because due to the presence of aquaporins along the transport epithelium that surrounds or makes up the wall of the loop of Henle. Okay. Nampak? Transport epithelium. So reabsorption of water continues through channels from by aquaporins, makes transport epithelium freely permeable to water. Remember, transport epithelium is the cells along the wall of the tubules. Okay. So movement is driven by the high osmolarity of the interstitial fluid, which is hyperosmotic to the filtrate. So tolong tulis, take note kat sini, kenapa dia cakap kat sini high osmolarity in the interstitial fluid. In this region, okay, surrounding the loop of Henle, you have interstitial fluid that is concentrated, that has, why is it concentrated? Because it has sodium chloride. It has salt in the interstitial fluid. Nanti kita tengok kenapa ada salt kat sini. Okay, so the more you enter into the kidney region, from the cortex into the outer medulla, into the inner medulla, the concentration of interstitial fluid will become more and more concentrated. It becomes hyperosmotic compared to the filtrate. Okay, so because due to the presence of sodium chloride, salt in the interstitial fluid, so that's why it uses the term hyperosmotic di sini. Hyperosmotic, lebih pekat berbanding dalam filtrate dalam loop of Henle kat sini. Okay, so because concentration outside is higher compared to the filtrate, so that's why more water will diffuse out, okay, through the aquaporins from the filtrate into the interstitial fluid. So there are almost no salt, no channel for salt and other small solutes. Okay, almost no channels for sodium chloride or other solutes to move out of the filtrate. Only water will move out due to the presence of aquaporin along the transport epithelium along the descending limb of loop of Henle. So the filtrate becomes increasingly concentrated. Nampak? Highlight kat sini. Increasingly concentrated sebab apa? Banyak air dah keluar kan? Air keluar. Tolong notekan dalam open diagram. Air keluar all the way through. So this will cause the filtrate to become more and more concentrated as it enters into the elbow of loop of Henle. Okay, so it says here filtrate becomes increasingly concentrated in solute concentration as filtrate loses water because the osmolarity of the interstitial fluid increases progressively from the outer cortex to the inner medulla of the kidney. Lagi awak masuk ke dalam inner medulla, lagi concentrated interstitial fluid, lagi banyak air keluar, lagi concentrate filtrate sampai dia enter into elbow of loop of Henle where the concentration is the highest. Okay, concentration or the osmolarity of the filtrate is the highest, which is around 1,200 milliosmol per liter of filtrate. Okay, kat sini at the elbow of loop of Henle. So this is the most concentrated osmolarity of the filtrate that reach at the elbow of loop of Henle. So dah sampai ke elbow, so dia naik balik dekat ascending limb of loop of Henle. Ascending, as you can see, for the ascending limb of loop of Henle, dia ada dua region. It has two region. So it has the thin segment of ascending limb of loop of Henle and the thick segment of the ascending limb of loop of Henle. Kat sini awak kena take note kenapa dia ada dua region yang berbeza, sebab dia berlaku dua proses yang berbeza kat sini. Okay, structure dia sangat significant where in the thin segment, okay, so this is where sodium chloride, arrow kelabu apa? Diffuse out. It diffuse out. It is diffuse out. It passively diffuses out from the filtrate into the interstitial fluid. So this helps maintain the osmolarity of the interstitial fluid in the medulla. Ingat tadi kan saya cakap inner medulla, as you go from cortex into the outer medulla, into the inner medulla, interstitial fluid concentration will become more and more concentrated. Sebab apa? disebabkan oleh faktor ni lah. Okay, dekat ascending, dekat thin segment, you have sodium chloride diffusing out of the filtrate. Sebab apa? Dekat sini tadi, you take note, kat sini concentration of filtrate is the highest, 1200 milliosmol. Disebabkan kat sini concentration filtrate sangat tinggi disebabkan oleh apa? Sebab banyaknya salt in the filtrate. Bila banyak dah salt dalam filtrate tu, dia akan menyebabkan salt tu keluar. Faham? Keluar hanya melalui proses passive transport. It passively diffuses out of the filtrate into the interstitial fluid. But as the filtrate moves up the loop, the ascending limb of loop of Henle, obviously less sodium chloride will be present. Tapi dia nak juga suruh keluarkan sodium chloride ni. Dia nak juga sodium chloride ni keluar ke dalam interstitial fluid. So, the transportation of sodium chloride at the thick segment is through active transport. Okay, so itu kena take note. Thin segment, passive, sodium chloride keluar. Thick segment, sodium chloride keluar melalui active transport. Sebab kat sini, concentration of filtrate becomes less concentrated. As more sodium move out, filtrate becomes less concentrated kat sini. Okay, so kat sini 1200, nanti dia naik, dia jadi 300 kat sini. Okay, 300 milliosmoles per liter. Okay, so take note please. So in the ascending limb of loop of Henle, salt but not water is able to diffuse from the tubule into the interstitial fluid due to the absence of aquaporin. So terbaliklah, ascending terbalik dengan descending. Descending ada banyak aquaporin, tapi kalau ascending there is no aquaporin, absence of aquaporin, so there will be no reabsorption of water, only sodium chloride will move out either through passive or active transport, depends on the segment. Okay, so the filtrate becomes increasingly dilute, okay, as it moves up to the cortex in the ascending limb of loop of Henle. So that's why as you move from the region of cortex, medulla and into the outer medulla and into the inner medulla, interstitial fluid ni concentration dia dekat cortex ni lagi less concentrated berbanding dengan inner medulla which is more concentrated. Sebab kat bawah ni lagi banyak sodium chloride diffuse keluar, ke atas ni hanya melalui active transport. Okay. So after the ascending limb of loop of Henle, it enters into the next region which is the distal tubule. Okay, distal convoluted tubule. Okay, convoluted is highly folded. So the distal, in the distal tubule, it regulates, okay, it controls the potassium ion and also sodium chloride ion concentration of the body fluid. Okay, so involves variation of the amount of sodium secreted maksudnya daripada darah, aliran darah, kan, peritubular capillaries that surrounds the distal tubule, secreted out from the blood vessels into the filtrate. Okay, into the filtrate, and sodium chloride reabsorbed from the filtrate enters into the interstitial fluid into the blood vessels. Okay, so like the proximal tubule, the distal tubule contributes to the pH regulation by the controlled secretion of hydrogen ion and reabsorption of bicarbonate ion. So bicarbonate ion it has to be reabsorbed from the filtrate into the interstitial fluid into the blood vessels, which is the blood vessels surrounding the distal tubule, which is the peritubular capillary. Okay, so next is the collecting ducts. Okay. Before I proceed, sekarang ni dah pukul 3, pukul 2. S10 sepatutnya ada tutorial dengan saya. S9 saya nak tanya, you all have class tak lepas ni?
Tak ada.
Boleh saya extend sampai 4:30 tak?
Boleh.
Saya extend, tadi pun kelas start lambat. Alright, so saya nak extend sampai 4:30, saya nak habiskan sampai tu solid model. Alright. Okay, so after the distal tubule, the filtrate enters into the collecting ducts. Okay, so the collecting ducts carries filtrate towards the medulla. Okay, towards the medulla region of the kidney and into the renal pelvis. Okay, ingat ya, renal pelvis tu yang structure corong tu lah sebelum dia masuk ke ureter. So water is lost as well as some salt and urea, and the filtrate becomes more and more concentrated. Kat sini dia dah dilute tadi. Kan, dia dah dilute. Sebab kat region kat sini banyak salt keluar. So bila dia filtrate reach the distal tube, dia dah jadi diluted. Tapi bila filtrate turun balik ke collecting ducts, because the transport epitheliums surrounding the collecting ducts here, they are many aquaporins. Okay, aquaporins. So along the collecting ducts, apa akan jadi? Water will be reabsorbed. Okay, water will be reabsorbed. So when water is reabsorbed, filtrate concentration will become more and more and more concentrated. And as you notice, start daripada Bowman capsule sampai distal tubule, tak ada urea kan keluar kan. So bila filtrate tu dah reach dekat inner medulla, due to the high concentration of urea in the filtrate, so this causes urea to diffuse out from the filtrate, okay, in the medulla region of the kidney. Okay, so urea and water will move out. Especially urea akan keluar in this region, in the inner medulla region. Okay, sebab sepanjang filtrate lalu dekat dalam tubul ni tak ada urea yang keluar, tapi dia akan keluar masa dia reach this region. Okay. So water is lost as well as some salt. Salt akan keluar melalui active transport. Okay. So water is lost and some salt and also urea, and the filtrate becomes more and more concentrated. The reabsorption or the moving out of sodium chloride and also urea, especially in this region, will cause the interstitial fluid in this region especially to be more concentrated compared to the filtrate. So kalau awak tengok dekat gambar rajah yang full kat sini, okay, cortex, outer medulla, inner medulla. Inner medulla will have higher concentration of interstitial fluid. Dekat cortex, interstitial fluid is not as concentrated as the inner medulla. Disebabkan oleh apa? Dekat region ni akan berlaku reabsorption of sodium chloride and also urea. These two solutes, urea and also sodium chloride, will contribute, okay, to the high concentration or osmolarity in the interstitial fluid. So the interstitial fluid will be more hyperosmotic compared to the filtrate. Boleh Boleh dengar bahasa mula? Interstitial fluid, concentration dia lebih tinggi berbanding dengan filtrate disebabkan apa? Disebabkan oleh urea yang keluar dekat region sini, urea keluar dan juga sodium chloride keluar. These are the two essential solutes that will make sure the interstitial fluid is always concentrated compared to the filtrate. So when you have a high or concentrated interstitial fluid, so this will make sure water will always and continuously reabsorb from the filtrate and back into your body. This is to make sure that water is conserved within your body. Okay, so these two substances yang sangat penting untuk make sure water will always continuously reabsorb from the filtrate into your blood so that your body will conserve the water and produces hyperosmotic urine, okay, or concentrated urine later, depending on the situation lah. So if your body want to conserve water, then more water will be reabsorbed. Okay. Alright. So sampai sini kan? Okay. So in the inner medulla, the ducts become permeable to urea because of the high urea concentration in the filtrate. Some urea, not all some urea will diffuse out. Okay. Sebab concentration of urea dah tinggi sangat dekat filtrate ni. right, in this region. So some urea diffuses out of the ducts into the interstitial fluid. Along with sodium chloride, this urea, tolong highlightkan, contributes, okay, to the high osmolarity in the interstitial fluid in the medulla. Please take note of this point. Okay, these two substances, these two solute, sodium chloride and also urea, will cause the interstitial fluid to be highly concentrated. It has a high osmolarity compared to the compared to the filtrate. Okay, so when producing dilute, ini tolong gariskan. Ini dah habis dah point untuk yang ni. Ini another new point. Kat sini sepatutnya new slide, sepatutnya kat sini. Ini untuk hasilkan concentrated urine lah. But if you want to produce diluted urine or hypoosmotic urine, the ducts will actively reabsorb salt and no water reabsorption, as epithelium will lack aquaporine channels and also sodium channel. Sodium chloride is actively transported out of the filtrate. Okay, so this is to produce diluted urine, to produce hypoosmotic urine. Kalau minum banyak air kan, if you for example drink a lot of water, so you will produce diluted urine. So this will cause you produce diluted urine disebabkan oleh, pada point kat sini, pada masa orang nak hasilkan dilute urine, there will be no aquaporine in this region, okay, in the collecting duct region, lacking aquaporine, okay, less or no aquaporine, less, okay. So sodium chloride will be actively transported out. Sodium chloride akan keluar, actively keluar. So this is to produce diluted urine. So fluid from several nephrons, nampak? Several nephrons kat sini flows into a collecting ducts, all of which lead to the renal pelvis, which is drained by the ureter. Maksudnya daripada ureter dia akan pergi kumpul kat dalam urinary bladder. Okay. So this is the whole diagram lah. So bila tengok gambar ni sekaligus kan, peningnya tengok gambar ni, tak kan? So kalau bila sesuatu diagram tu dibagi selambak macam ni, analysekan dia satu-satu. Okay, tengok daripada point nombor 1, nombor 2 apa jadi kan, nombor 3 thin, thick segment, and then distal tubule, and then collecting ducts. Okay, so on your own nanti, okay, please look back to this diagram, okay, fahamkan proses ultrafiltration, reabsorption, secretion, what happened in this region, what happened in the descending limb of loop of Henle, where you have reabsorption of water. Why there is reabsorption of water? Because the interstitial fluid is much more concentrated compared to the filtrate. Why is it more concentrated? Due to the reabsorption of these two solute, urea and also sodium chloride. So as more water is reabsorbed down the limb of loop of Henle, kan? So filtrate becomes more and more concentrated. Maximum dekat sini 1200 milliosmol per liter. So bila dia naik, filtrate jadi more and more concentrated, right? So sodium chloride akan diffuse keluar. Tapi bila dah reach dekat thick segment, dia nak keluarkan juga lagi. So it is melalui active transport. Okay, so dekat region ni, because you remove sodium chloride from the filtrate, sodium chloride is being reabsorbed, so filtrate becomes more and more dilute. Kan? So region yang kuning kat sini adalah region where concentration of filtrate is diluted. Kat region kat sini, concentration of filtrate is around 300. Okay, 300 region kat sini, right? So kat sini akan berlaku apa? Reabsorption, secretion, and then enters into the descending limb. Okay, so this is where reabsorption of water occurs. Sodium chloride reabsorbs, urea reabsorbs, you produce hyperosmotic urine, concentrated urine. Okay, vice versa, if you want to produce dilute urine, so there will be no reabsorption of water. Okay, you produce diluted urine and you are going to move sodium chloride out of the filtrate. Okay, so balik nanti, bukan balik maksudnya after lecture, tengok balik diagram ni and fahamkan balik proses apa yang berlaku kat setiap region. Okay, so okay, so solute gradients and water concentration. So it says here, urine is much more concentrated than blood, which is about 1,200 milliosmol. This is when you are producing a very concentrated urine. Okay, hyperosmotic urine. So the blood concentration kat sini is blood concentration is around 300 milliosmol. So this is the concentration of blood. This is the concentration of urine. Okay, tolong betulkan balik. Letaklah full stop kat sini. Urine is much more concentrated than blood, which is about 1,200 in the urine, and in the blood its concentration is about 300 milliosmol. Okay, so as I've mentioned and I've explained about the juxtamedullary nephron. Okay, juxtamedullary nephron extend deep into the medulla that are essential for the production of urine that is hyperosmotic to body fluid. So juxtamedullary nephron is really important to conserve water within the body because it has a longer loop of handling. So when it has a longer loop of handling, more water will be reabsorbed compared to the cortical nephron where it has only short loop of handling. So it does not really reabsorb water so much as the juxtamedullary nephron. So dia punya application dia, it is beneficial for the body to conserve water, especially for animals that live in a hot area. So for this animal, they want to conserve water, so they will produce very little volume and concentrated urine. Okay. Okay. So solute gradients and water conservation. So the cooperative action and the precise arrangement of the loop of Henle and the collecting ducts are largely responsible for the osmotic gradient that concentrates the urine. Okay, so kena fahamkan kenapa saya emphasizekan pasal region-region tu tadi. Outer and also the inner medulla region. The presence of sodium chloride and also urea tu. So these are the important component that plays the role in producing concentrated urine and to maintain a balanced body fluid concentration. Okay, so these two solutes, sodium chloride and also urea, okay, contributes to the osmolarity of the interstitial fluid, which causes reabsorption of water in the kidney and concentrates the urine. Okay, bila ada banyak solute, especially sodium chloride dengan urea dalam interstitial fluid surrounding the loop of Henle, more water will be reabsorbed and you will produce concentrated urine. Okay. Okay, so that's lead to this model which is two solute model. So ini awak kena fahamkan betul-betul proses tadi dengan two solute model ni. Okay, so it is important in producing in concentrating urine lah in mammalian kidney. So it says here, as you can see from the diagram here, the osmolarity of the blood, which is 300, is the same as the filtrate in the proximal tubule and also in the distal tubule, which is around 300. Okay, 300 milliosmol. So in the proximal tubule, the filtrate volume decreases. Sebab apa? Water is being reabsorbed. Ingat balik gambar tadi, water is being reabsorbed, but its osmolarity remains the same. Okay, as the blood. Okay. So filtrate passing from the Bowman capsule in this region here, okay, in the Bowman capsule to the proximal tubule has an osmolarity of about 300 milliosmol, the same as the blood. Okay, so large amount of water and salt is being reabsorbed from the filtrate as it flows through the proximal tubule into in the renal cortex. This is the cortex region of the kidney. So tolong tulis kat sini, jot down. Okay, so you have reabsorption of water keluar, and also sodium chloride keluar. So this maintains the filtrate at 300 milliosmol. Okay, so next, as the filtrate flows down, so filtrate tadi kan, kat proximal tubule, dia punya concentration is 300 milliosmol. So as it goes down the descending limb of loop of Henle, water is being reabsorbed all the way through because there is aquaporins in the transport epithelium of the descending limb of loop of Henle. So water is being reabsorbed continuously. So when water is being reabsorbed continuously, water move out of the filtrate into the interstitial fluid, into the blood vessels surrounding the loop of Henle. Okay, so as water moves out, the concentration of filtrate nampak kan daripada 300 turun jadi 400, 600, 900, 1200, which is the maximum concentration, okay, in the filtrate. So as the filtrate flows down the cortex to the medulla region, in the descending limb of Henle, water leaves the tuber by osmosis. Osmosis mesti melalui aquaporin. Ingat osmosis has to go through aquaporins. Okay. It involves what passive transport, which is to be specific, is facilitated diffusion because it is through aquaporins. Okay, so solutes including sodium chloride becomes more concentrated, increasing the osmolarity in the filtrate. So disebabkan air banyak keluar, air keluar. So filtrate jadi pekat. Disebabkan oleh apa? Dekat region ni tak ada pun sodium chloride yang keluar. So it causes the filtrate to be very concentrated, especially due to the presence of salt, okay, which is sodium chloride. So as the filtrate move up, so sodium chloride akan keluar melalui passive transport in the thin segment. Okay, thin segment is through passive transport. Okay, sodium chloride diffuses out of the filtrate. So it says here, the highest osmolarity in filtrate occurs at the elbow of the loop of Henle. This maximizes the diffusion of salt, okay, out of the tubule as the filtrate rounds the curve, kan, dia naik and enters into the ascending limb, which is permeable to salt but not to water. So ingat kat sini hanya garam je yang keluar. Passive transport sebab filtrate is really concentrated. Active transport dia nak keluarkan juga lagi salt tu. Okay, so this helps maintain a high osmolarity in the interstitial fluid, especially in the inner medulla region. Okay, so it says here, the loop of Henle has several qualities. Apa quality dia tu? Okay, which is of counter current system, which involve counter current. Ingat balik dia punya definition, the flow of fluid in opposite direction, right? So fluid in the filtrate flows in the opposite direction between the descending and also ascending, kan? Okay, so the loop of Henle expend energy to actively transport sodium chloride. So that's why kita panggil sistem ni sebagai counter current multiplier system. Because it uses energy. The process expends energy to make sure that more sodium chloride move out into the interstitial fluid from the filtrate. Okay, so that's why we call it as counter current multiplier system. It uses energy, okay, to move sodium chloride from filtrate into the interstitial fluid. So from the filtrate in the upper part of the ascending limb of loop of Henle, sepatutnya, tolong betulkan kat nota awak ni, sepatutnya this is one continuous sentence sebenarnya. Okay, kita baca balik semula. Loop of Henle has several qualities of counter current system which involve the loop of Henle to expend energy. It uses energy because it involves active transport to actively transport sodium chloride from filtrate from the filtrate in the upper part of the ascending limb of loop of Henle. This is actually one complete sentence kat sini. So please remove this full stop, sambung dan dia kat sini. Buat arrow je lah. Okay. So this maintains a high salt concentration in the interior of the kidney, enabling the kidney to form concentrated urine. Okay, concentrated urine. Sorry, tadi saya salah bukan 300 kat distal 100 je. 100. Kat sini baru 300. Tolong betulkan facts tadi. Alright. Okay, so this filtrate is hypoosmotic to body fluid by the time it reaches the distal tubule. Nampak? So the concentration of filtrate is really diluted, which is around 100 milliosmol. Hypoosmotic. It has less solute concentration and more water. Okay, hypoosmotic to body fluid. Body fluid, interstitial fluid is 300. Outside is 300. Keluar ni, interstitial fluid is 300. But the filtrate is really dilute, which is around 100 milliosmol. Okay. So, explaining on the vasa recta part, okay. So the vasa recta is, as I mentioned, it is the blood capillaries that surround the loop of Henle, okay, of juxtamedullary nephrons. Okay, of juxtamedullary nephrons. So for vasa recta dia ada dua region juga, sama juga macam loop of Henle. It has the descending and also the ascending vasa recta. Dekat region yang mana loop of Henle descend, okay, vasa recta akan make sure darah tu naik ke atas. Kat mana loop of Henle naik, okay, vasa recta akan turun. Macam itulah. Okay, so this is significant dalam memastikan interstitial fluid is maintained at the right concentration, and also urine produced is also maintained at the right concentration, which is it helps in the production of concentrated urine. Okay, so again, saya tak suka awak refer gambar ni, it is a bit confusing, so please refer to this one. So this one is much clearer. Okay, so for vasa recta it has two regions, descending and also ascending vasa recta. Descending vasa recta dia surround loop of Henle yang ascending. Okay, kalau ascending vasa recta dia akan surround loop of Henle yang descending. Okay, so this is counter current juga. This is counter current sebenarnya. So the flow of the filtrate in the descending here, descending limb of loop of Henle, it flows in the opposite direction as blood in the ascending vasa recta. Nampak tak ni kat sini? Okay. Sama juga flow of filtrate in the ascending loop of Henle is in the opposite direction as blood in the descending vasa recta. Tengok dia punya arrow kat sini. Okay. So it says here, what prevent the capillaries of the vasa recta from dissipating the gradient by carrying away the high concentration of sodium chloride in the medulla's interstitial fluid. Okay, so so dengan adanya arrangement ni, dia akan make sure sodium chloride concentration or interstitial fluid concentration is high outside, and water will always be reabsorbed. Okay, this arrangement, it makes sure of that. Okay, so it says here, as the descending vessels convey blood towards the inner medulla, water is lost from the blood as sodium chloride is gained by diffusion. Tolong tulis dekat nota awak kat sini. Sebab saya dekat desktop saya susah nak tuliskan dia. Okay, darah kan? Darah bila dia lalu region dekat sini, dia akan menyebabkan air dalam darah ni keluar. H2O keluar. H2O keluar. Okay. H2O keluar. Kenapa H2O keluar? Sebab awak fahamkan balik dekat ascending limb of loop of Henle, apa yang berlaku? Yang akan terjadi adalah salt will be reabsorbed. Salt akan keluar. NaCl kan, dekat ascending kan ada NaCl keluar. NaCl keluar. Okay, bila NaCl keluar dekat ascending limb of loop of Henle, concentration automatically concentration kat sini, interstitial fluid punya concentration lebih tinggi berbanding concentration dalam darah. Bila concentration interstitial fluid dekat dalam interstitial fluid tinggi, so dia akan menyebabkan air dalam darah tu keluar. Air dalam darah keluar, air dalam darah keluar. Faham? So it says, itu yang point kat sini. As descending vessel conveys blood towards the inner medulla, dia turun kan. Water is lost from the blood as sodium is gained by diffusion. Sodium chloride yang keluar daripada ascending limb of loop of Henle, dia keluar masuk interstitial fluid dan juga dia masuk ke dalam blood vessels tu. Okay, dia masuk dalam blood vessels. So blood vessels dia carry blood daripada descending pergi pula kepada ascending. So ascending blood vessels dia akan flow in opposite direction as filtrate in the descending limb of loop of Henle. So ingat balik, kalau descending apa yang jadi? Air keluar kan? Air keluar, NaCl, H2O keluar. So bila H2O keluar, region kat sini sebenarnya akan jadi dilute kat sini kan, sebab air keluar daripada distal tubule. Bila air keluar daripada descending limb of loop of Henle, air tu akan masuk balik dalam darah awak. Tadi air keluar daripada darah kan, ni air masuk balik dalam darah. That is reabsorption. Okay, reabsorption of water. So as water from descending leaves the filtrate and then it enters into the blood vessels, darah tadi yang mana dia ambil NaCl dekat region ni, dia akan keluarkan balik dekat region ni, NaCl. Faham ke? Okay. So net flux are reversed as blood flows back towards the cortex in the ascending vessels where water re-entered the blood and NaCl diffuse out of the blood vessels. Okay, so vasa recta able to supply the kidney with nutrients without interfering with the osmolarity gradient in the inner and outer medulla. So bila darah naik kan, so darah akan ambil air daripada descending limb of loop of Henle, and at the same time kan dia akan bagi nutrien lah juga, nutrien kepada kidney. Okay, it gives nutrient to the kidney. Alright. So it says here the counter current like characteristics of loop of Henle and the vasa recta helps to generate the steep osmotic gradient between the medulla and also the renal cortex. Saya nak tanya sekejap, semua orang clear tak dengan apa yang saya bebelkan ni? Faham ke?
Clear.
Clear, madam.
Boleh ya? Alright. Okay, so last slide. Alright. So concentrating urine in the mammalian kidney, again still on the two solid model. So from distal tubule is this region here. Okay, so the filtrate descend towards the medulla, this time in the collecting ducts. Okay, so the collecting ducts conduct filtrate through the osmolarity gradient. So interstitial fluid, as it enters from cortex into the inner medulla, the interstitial fluid will become more and more concentrated, hyperosmotic. Okay, so as filtrate passes cortex to medulla, it encounters interstitial fluid of increasing osmolarity. Okay, so this results more water to exit. Nampak? Lebih banyak air keluar. Sebab apa? Concentration interstitial fluid kat interstitial fluid ni tinggi, alright? So this result more water exit the filtrate by osmosis and concentrating salt, urea and other solute in the filtrate. So urea at this point will diffuse out from the filtrate in this region, in the inner medulla region from the collecting ducts. So this contributes to the high interstitial osmolarity of the inner medulla. So these two solute, sebab tu kita nama proses ni sebagai two solute model, disebabkan oleh dua solute ni, urea and also sodium chloride. forms the osmotic gradient that enables the kidney to produce urine that is hyperosmotic to the blood. Okay. So saya just nak draw again kat sini, kat gambar ni to make it much more clearer. Good. Okay. So, bila awak tengok gambar raja ni yang saya cerita pasal vasa recta tu, awak tengok blood vessel saya draw dia sebagai warna merah. Okay, so this is vasa recta. Okay, so this is also vasa recta. Tolong labelkan saya ini apa? Vasa recta apa?
Ascending.
Ascending. Okay, ascending vasa recta. Yang ini?
Descending.
Descending vasa recta. Ascending vasa recta is when blood flows in the opposite direction as the filtrate. Blood flow ke atas, naik balik. Descending, blood flows ke bawah, opposite direction as the filtrate in the ascending loop of Henle. So tengok bila darah dia turun kan dekat descending limb of loop of Henle, nampak kan sodium chloride ni keluar daripada filtrate. Sodium chloride yang keluar daripada filtrate ni, kalau nak mudah nampak dia sebenarnya dia masuk juga dalam vasa recta ni kan. Dia keluar daripada filtrate, masuk interstitial fluid, masuk ke dalam vasa recta, descending vasa recta. So darah daripada descending vasa recta ni akan kehilangan air. Dan akan kehilangan air. So bila dia naik balik ke ascending blood vessels, so dia akan regain baliklah air yang dia lost tadi. Faham tak? Sebab bila dia lalu dekat sebelah descending limb of loop of Henle, akan berlaku reabsorption of water. Salt yang dia absorb dekat region ni tadi kan, so dia akan keluarkan balik. Faham? Dia akan keluarkan balik salt tu. So this will maintain and make sure that the sodium chloride concentration is high, especially in the inner medulla region. So in the inner medulla region, its concentration, the interstitial fluid concentration is high due to this solute, urea and also sodium chloride. Okay, so that is two solute model and counter current dan juga counter current multiplier system tu tadi. Counter multiplier sebab apa? Dekat region kat sini dia melibatkan active transport of sodium chloride that expand energy. It uses energy. That is multiplier. It expands energy. Okay. So next one yang ni kita akan tengok esok lah kot. Okay. Tomorrow we are going to proceed to these two hormones, and also we are going to look at the next chapter, the new chapter, which is on reproductive system. So make sure esok, tomorrow, you have your lecture note lah for repro. Okay, for repro. So the lecture note repro boleh download dekat YouFuture, and make sure